Hi, this is Terry Kuzma here at Penn State University, and what we're going to look at today is part 3 in our series of bottom-up manufacturing, and we're looking at surface wetting and adhesion. And today we're going to take a look at adhesion and adhesion in the liquid state. So we're looking at examples that would promote that type of interface. So what we're again is we're looking at material interface forces here in part 3. So interaction in the liquid phase, not only are liquid to surface interactions important, as we viewed with the gecko foot, for example, and the synthetic gecko tape that people make, but liquid to liquid interactions also can be considered. So the hydrophobic interactions describe the relationships between water and hydrophobes, and these are low water soluble molecules. Hydrophobes are nonpolar molecules and usually have a long change of carbons that don't interact with water molecules. At the molecular level, the hydrophobic effect is important in driving protein folding formation of lipid bilayers and of myocells, and insertion of the membrane proteins into nonpolar environments and protein small molecule interactions. So we took a look at a lot of these issues within the presentation that I had on cancer and cancer drugs that involve liposomes. Because of the inherent efficiency of material arrangement due to its affinity, this can also be seen in nature, and certainly can be seen in nature because this is an efficient use of, I don't know to say energy because it takes very little energy for this to take place, but it's an efficient material interaction network. So let's take a look at some more definitions and look at molecules in detail and divide them into two substates. So we can look subsets. We can take a look at polar molecules. This is where electrons are not equally shared. One part of the molecule is more negatively charged than the other part. These are naturally hydrophilic and bond to polar water structures. So nonpolar molecules, these electrons are equally shared. The molecules don't have poles specifically on them, and these are naturally hydrophobic and avoid interaction with water. So we can take a look at examples of these. We can look at a nonpolar molecule, and I guess the classic example here is carbon dioxide, and where we have the opposite charges of the negative charge on the outside, canceling themselves out, so it doesn't have a net charge to one end or the other. If we would look at the polar molecule here in the B example, we can see that there is a charged anomaly here, that one side is more positive and the other side is more negative. So the negative charge would be pulled more towards oxygen. So again, this is important in the bonding of water and the attributes of how water has such a high, relatively high, its boiling temperature and its freezing temperature, and the expansion and contraction of the liquid. So let's take again more look at this interaction in the liquid phase. The mixing of fat and water is a good example of this particular interaction. A common misconception is that water and fat don't mix because of van der Waals forces, but this is not really true. The forces that are acting upon both water and fat molecules are too weak to have this. This immiscibility can be used as a patterning technique, and we'll look at this as an example, and we have throughout the course in a few areas of block copolymers. So what we have to do to define what a block copolymer is we have to look at polymers in general, divide that, and show what a block copolymer is. So a homopolymer is the same across its building blocks. A random copolymer has two different building blocks, and those again are randomly associated in their linkage. An alternating copolymer, like the name says, as we can see in the example, alternates between the two different types of polymers. And then block copolymers, which are found in nature quite a bit and studied in materials such as proteins or protein networks. This is a polymer that has one end is one type and the other end is a different type of polymer. So that's shown in our example down below, where you have block A, and then there's a transition and there's block B. So this is a one big macromolecule, and basically it's linked together, and one side is unique compared to the other side. So let's take a look at this interaction in the liquid phase. Basically, if we would take this material and we dissolve it together, these polymers, the most famous block copolymer for lithography patterning, and that's what we took a look at in when we had a discussion about uh, growing nanowires. We would use this methodology to pattern the catalyst. So this would be our resist to do that work. So probably the most famous block copolymer per se for uh, lithography work is the PS PMMA interface. So this shows us schematically down at the bottom that we have the PS block, which is represented by the chemical association here on the left in blue, and then we have the PMMA block. So basically what we can have is different ratios of PS to PMMA. But this is what we can do is we can take this material, this basic unit, and then when you mix it together on the macro and spin it, it's going to separate in an energetically favorable condition. So depending on the ratio to PS to PMMA, if we would have example here, a 20 to 80 ratio, or a 30 to 70, 50 to 50. So this is the increasing length of A as we go to the right. So A is shown as 20. And as we go to the right, it goes to 30, 50, 70, 80. So when we take a look at this and we do a formation, a spin, so this is a liquid, we spin this on a substrate, allow this to reach a thermodynamic equilibrium. And what we're going to do is form these specific patterns. So these patterns are indicative, again, of the ratio of PS to PMMA. And now we can use this patterning to our advantage because it has self-formation, and basically it takes very little infrastructure or energy to make this. So it's basically, we would pour this on the substrate, do a spin, and then potentially an anneal. So this shows the interaction of the material and the phases. So we took a look schematically at what they look like, but here is material that we generated here at Penn State University. So this is done by a colleague of mine, Travis, who's no longer at the university, but certainly a brilliant researcher. He actually created these block copolymers and took these pictures, and it shows us the ratios of these block copolymers and what they actually look like. So again, schematically, we took a look at that, but these are the actual diagrams of materials that we processed in our laboratory. So this shows us a 30-70 mix of PMMA to PS, and we get these small holes. Again, these holes are going to be the 30 part, right? And then when we do a 50-50, we get this pattern described as lamellae. And to me, it looks like a fingerprint. So we have these random channels throughout the material. And then when we invert the amount, the ratio of PMMA to PS, and we do a 70-30 mixture, what we basically get in here is a mirror of image. So this shows the ratio of the materials and the process that we make. So again, I'm going to go back in the slide here and say, schematically, artistically, this is the drawing that we were looking at. But in, for a real example, these are pictures of materials that we processed in our lab, and Travis actually looked at with our FESEM. So this is one of the reasons that we would like to use, per se, different types of copolymers. And one of the bigger reasons to use a certain types of block copolymers is certainly the affinity to adhere to each other, but it's also the affinity because at some point when we're using it for lithography, we're going to want to get rid of or volatilize one component of that block copolymer. So for example, what we would like to do is etch away either PS or PMMA. So this shows us the etch selectivity. So if we would look at materials such as the PS PMMA, we have the etch rate here on our y-axis, and on our x-axis we actually have the measure of carbon and oxygen content in one polymer. So this is a ratio. But if we would actually look at and maybe focus on the etch rate, if we have a material like PS right here, it has a rather low etch rate. And then if we would look at, so I'm focusing right here where we have this PS block right here, this circle. And then if we would look at PMMA, PMMA has a tremendously high etch rate. And so now if we would process this material, and let me go back in this diagram, and we have this PS PMMA matrix, what we could basically do now in this area right here, we could, with a 70-30 mixture, we could etch the in a plasma that's discussed here, and the plasma relationship that would have for maybe a CF4 oxygen mixture. What we can do is etch in there, and what we would leave is the PS on the surface, and the PMMA would be removed. So basically what we're trying to do in some senses is like an exposure and development in conventional lithography. But in this method, what we're going to do is we basically use the enhanced etch rate of PMMA to be able to remove that selectively with respect to PS. We can go further in that construct, and I don't have a slide in here, but basically what we can do is a process called staining. And in staining, what we could do is apply another material after we have the block copolymer formed. So we could mix other materials and actually reduce the etch rate of PS and move it down into this region. So again, with further processing, we can shift the relative strength of PS, and we are able to lower that on a chart. Again, the goal here is to have a higher selectivity when we do that etch process so that we could form openings in our materials, for example, to do work like a liftoff process. Probably one of the bigger disadvantages, let me go back in the slides, for these block copolymers, we actually have to apply the thickness of this block copolymer according to the chain length. So this chain length is typically on the order of, say, 40 or 50 microns maybe. So that's the relative thickness or thinness, however you want to look at it, of the material that we spread across the wafer. So to have these well-formed geometries, what we're going to have to do is only be able to have this height on the order of 50 nanometers or so. And we would also have to have an additional randomization layer on our surface so that these don't preferentially treat. So again, we look at those details of this process in detail in our, I'm taping, I'm sorry. So I'm sorry, the phone rang there. What we're going to do is look at this in detail, and we would have a randomization layer here. Again, I think Oscar does a good job in his course of review of lithography materials, and I discuss this quite a bit, I think, in the course on growing silicon nanowires. So I'll leave it to more detail in there, but you can always ask me questions too. I just wanted to make sure that we cover that. So these, when we look at this interaction of hydrophobic, hydrophilic materials in a liquid state, this is a really good unit, or a really good tenet to use in biology, because we have to minimize our energy, and these things tend to be really predictable. So these types of networks we can see in nature are found in cell membranes and in proteins and in DNA. So these themes that we would envision and see that biology does, we can actually copy those and take them into production, or understand them so that we can better make drugs or products, etc. So, or prosthetic devices. So let's take some examples of that. So one of our favorite structures that we've looked at multiple times throughout the nanotechnology course is the phospholipid. So naturally we have this phosphate head, and this head is hydrophilic, and this head tail is hydrophobic. So what we're going to do is use this formation later on to take a look at self-assembly units. So what is a phospholipid? We looked at this before. It's a natural self-assembly unit. It's a classical example of surface interaction. The phospholipid is a molecule related to fat. It has two heads, two sides compared to a hydrophilic water-loving phosphate head, and a hydrophobic water-fearing fatty acid tail. When the phospholipids are added to water, they can then self-assemble. The phosphate head points towards the water, keeping the fatty acid tails away from the water, forming a bilayer. These bilayers always actually are used in nature to form cell membranes. So this is, an example of how we copy nature, and we took a look at this in detail in our discussion about cancer and man-made nanoparticles, specifically the liposome. But this is for a review, an example that this is a drug called Doxil, and basically it's a passive delivery drug. And what we do is we take a fat-containing substance, a natural bilayer material, such as an egg derivative. So chicken egg derivatives is where we would get this complex from. What we would do is form a cake out of this, a solid matrix, and then when we mix it together in an aqueous solution, and the aqueous solution could have an entrapment of a drug in it, such as doxorubicin HCl, and what will happen in that form, when we add water, these lipid bilayers are going to form a thermodynamically stable formation network, and they're going to form a sphere. Inside that sphere, what they're going to do is trap this aqueous liquid. The aqueous liquid is going to have our drug in there, and so now what we've done is made a formation of this sphere with a drug on the inside. So now this lipid bilayer is going to act to protect the drug when it's in the body, and then we see this outer core, these things that look like hairs, they're actually going to be a sugar coating, a polyethyl glycol coating, and what they're going to do is provide protection from the removal or attack system of the body. So now we can insert this drug inside the body, and what it's going to do is have a relatively long half-life on the order of a few days, and it's going to circulate in the body, and it so happens, due to the size of this nanoparticle, probably around 100 nanometers, 60 nanometers, in and around there, depending on the type of drug that it is. There's multiple diameters of this drug in this analogy. This is going to be able to fall into the defects that are indicative of areas where there's cancer or tumor formation. And in that way, when it goes into that area, the cancer is going to take in this drug, break it down into food, and when it does, it's going to release the drug inside the cancer. So I guess this is a formation of an active drug or a passive delivery of drug to kill cancer. And this shows us the animal cell, and again, the lipid bilayer here is formed in the nuclear wall, and it's also formed in the cell membrane itself. So we use this hydrophilic interaction in a lot of places around the cell. Again, nature, I guess the currency of life, most people would say, is proteins. So this is the DNA giving us a subset of RNA, and what the ribosome does is rides along these data interfaces called codons, and what it's going to do is assemble material called amino acids, and these amino acids, when they're folded in an aqueous solution, are going to give us this hydrophilic interaction to give us these complex three-dimensional protein structures. So this again is an example in our discussion for this part in the presentation of hydrophilic interaction within an aqueous media. So this is an example of that, and what we use is nature to provide these details and how it's done. So this shows us the interaction of the lipid bilayers and how it forms the cell membranes in here. So we can see this, and it's interesting that the cell membranes are held together by cholesterols. These are cross linkers. I think that it's shown right here. Yes, this yellow section right here is cholesterol. So there's good and bad cholesterols. In this case, these are good cholesterols because they're giving us durability to cross-linking for these cell membranes, the lipid bilayer in here. It helps make them more durable. So in our analogy, when we're making a liposome drug, if we want to make that liposome more or less robust, the cell membrane on the, or not the cell membrane, but the membrane of the lipid bilayer, to make that more or less mechanically robust, what we're going to do is use cholesterol as cross linkers. And again, what we're going to see is in this cell membrane, we have these conduits or surfaces like this protein transport channel. What we do is basically in nature, we would take another protein, send it over into this area. When it couples with that area in its complete three-dimensional architecture, it'll elicit a response that we can open or close this transport channel. So basically, this is a lock and key network that we use protein interaction between cells to elicit response and to get some kind of functionalization of these cells to be realized. So this shows us the lipid bilayer, another brilliant technique that we use within this lipid bilayer. For gases, the lipid bilayer is actually permeable to gas like carbon dioxide and oxygen. So this reduces the need for the cell to actually perform this task and use energy. It's naturally transparent to these gases. We can also take in hydrophobic molecules such as benzene. That could be good or bad. One of the reasons it's bad and a carcinogen is that the membranes don't protect the cell from this. So this benzene could go through this membrane, and it could also go through the membrane that's the nuclear envelope and cause problems for, say, with rogue interactions with the DNA. So that's one of the reasons that benzene, we can see now, is carcinogenic. We can also see that small polar molecules such as ethanol and water are also transparent to this cell membrane and be able to move through there with very little uptake in energy. But other molecules that would certainly impact the cell are not directly transferable through the lipid bilayer immediately. We can have like endocytosis, which sometimes is receptor mediated, that we would have the cell membrane be able to engulf materials such as glucoses and take them into the cell, or they might be actually taken through receptor portals such as this protein channel, or it could be another receptor channel. So we can have these channels that could be opened through a protein interaction to take materials in, and certainly amino acids could be taken in through these other portals themselves. So these are charged molecules. They don't like to be directly transported through the membrane, so that we would have to use, per se, conduits and channels like this that would be regulated through protein signals to take in these particular materials. So again, these are fairly taken in by the cell without, per se, macro regulation, and materials like this have to be regulated in there. And again, our liposome, when we go back here, this liposome is going to appear to be a sugar. So generally these are taken through some type of channel or through endocytosis. So doxal appears to be a glucose on its outer core, so it directly will not go through this cellular membrane. What will have to be done is we might have to further tether some receptor mediated reaction on there, such as some kind of ligand, and that might enhance endocytosis, or we can take that through a particular channel. So this is the again, a really nice example. I really wanted to show this within this presentation. Mostly, I don't know, I guess, to give credit to nature, because this is really interesting. I think that this is a brilliant analysis and a look at what our colleagues have done in biomaterials to take a look at how we have these interactions of these molecules on the cellular level. So I think these are important for, certainly understanding diseases, et cetera, and drug treatment. So I think that this is a really nice slide. So interaction in a liquid state, proteins are certainly a major class of biomolecules, and these directly connect biology to nanotechnology. And again, another one of my associates here at Penn State, Abden, he did a lot of work with adhesion materials like glues that were basically, he saw these types of networks developed in proteins that exist in squids. And I thought that that was really brilliant work that he did. So understanding these biomolecules and being able to replicate them as man-made molecules are really important. So the adhesions or adhesives that he looked at and the applications for biomedical materials are really very nice. I think I'll put some links to his papers and researches in our course materials, and you can take a look at them. I actually thought he did really, really good detailed work. So our bodies themselves are about 20% protein, and what these proteins do is allow us to think, feel, move, and function, and naturally these proteins have to be correct. So if we would have damage to our DNA or RNA in the body, then we're not going to make the correct proteins, and then that's not going to allow us to think, feel, and move and function well. So again, this is what we have to do, is maintain this look at the importance of DNA, RNA, and proteins within the body. So these proteins themselves give many functions in the body. They act as catalysts for reactions. They're cellular gatekeepers, as we just discussed. They help with the immune system. They help monitor and the recognition of foreign cells and foreign objects within the body. And they, as we've seen before, that the proteins act as support for cells and tissues that we looked at in the lipid bilayer a little bit ago. And these proteins are very, very complex. Not that DNA is simple, but DNA is one structure, and it has one basic function to be a template for RNA, but we see that proteins themselves have a myriad of different interactions that they perform within the body. And again, these interactions occur certainly through the hydrophilic, hydrophobic interactions of the building blocks, which are amino acids of the proteins, and the aqueous solution in the body. So this is certainly our discussion for today, is the hydrophobic, hydrophilic interactions of these biomolecules in the body. So proteins are built from 22 types of amino acids, and these amino acids are hydrophobic and hydrophilic. So the DNA provides this template. These proteins themselves can act as binding sites for chemical reactions. These proteins are used for cellular communication, and these proteins are exhibited or expressed by cancers. So I know that for particular types of cancers, what they would do is take a look in your bloodstream, etc., and they would dissect your body fluid, blood areas, and look for specific proteins, and these proteins would be a clue to different types of cancers in the body. And we can also use proteins to coat materials such as catheters before they insert them in your body. And what we're going to do is, when we insert these catheters or materials in the body, we're going to elicit a certain response from the body. So these are, again, the hydrophobic occurring amino acids. So this is a list of the amino acids that are hydrophobic. So these are going to try to bury themselves inside the protein core. And then these are the polar amino acids that participate in hydrogen bonds. And these are very important to see, and they're often they're polar uncharged side chains. And then these are the charged side chains, and these often make salt bridges within the body. So these are the positive chains, these particular amino acids, and these are the negative side chains in the body. And these are special cases of these different types of amino acids. And again, these show the tendency to form these complex proteins protein units within the body due to the interaction of these materials within the aqueous solution. So here's the hydrophobicity scale for amino acids. So these are the particular amino acids in the body, and this shows the amount of hydrophobicity in those particular systems. So these are the researchers and the documentation of these particular amino acids. So this just shows the folding that we discussed before in other courses. We have this linear amino acid strand. When we form it into a liquid, what it's going to, or into water, it's going to have this hydrophobic hydrophilic interaction, and it'll form these complex structures. And often we might have more than one of these amino acids interacting, which we would have certainly a tremendous macro structure made out of a multitude of amino acids chains to give a tremendously complex system. So these are interactions in a liquid phase. We can see what, the different types of things in the body that we have. We can see, for example, this is actin fibers within the body, which help hold things together. We can see what transfer RNA looks like, ribosomes, etc. So these are different formations, and we can see the tremendous complexity of these materials. And for the most part, just relatively speaking as a commentary, these things are tremendous and tremendously complex, and I'm in awe of the complexity of protein and biology because it's so vast. And I think this stuff is mind-boggling, especially looking at this type of protein and the interfaces that this must have. Certainly compared to the simplistic stuff which I've spent a number of years studying, such as semiconductors and solid-state physics, etc., which is certainly intriguing and complex, but there's a certain rules associated with that, and it's a rather simple, quantifiable thing. This stuff here, this biology and these protein interactions, they're tremendously huge in their interaction and their ability to make bonds. So this stuff is tremendous. So one reason that I think that biology is a lot more, per se, challenging than biomaterials, I think that's true. So again, let's take a look at interaction in the liquid phase. The cell's membrane lipid bilayer has many receptors, which receive chemical signals from other cells in the extracellular matrix. A ligand is a molecule that binds to a receptor in order to produce a cellular response. So a ligand can be defined, it's a protein. It is a neurotransmitter, a hormone, a pharmaceutical drug, a toxin, a protein, or other cells or other membranes can induce these responses. So proteins on the cell membrane serve as markers to identify one cell to another in a specific cellular environment. And we had a discussion when we looked at cancer and liposomes of recognition of like red blood cells and bone cells in the body. So we have to look at these issues when we want to develop materials such as prosthetic devices, such as screws that we would put into arms when they break, or knees, or artificial hips, or dental implants. So these materials, man-made materials, must emulate the body. So often what we would do is control the hydrophobicity of the material so that we could control water and protein interaction on there, because once we tether proteins and sugars onto a man-made material, we can control its biological response or bioavailability. So this shows the wetting of proteins and cellular growth. So this shows, again, what happens with a membrane and how we would pass materials between themselves. We show what an enzyme would do. These pictures also, again, these are things that we reviewed in other presentations, but these show the roles of proteins and what they do. We can have the surface cell identity markers. We can have cell surface receptors, and certainly these proteins help in cellular adhesion, such as tight junctions, which we took a look at when we defined cancer as not having good cellular adhesion or tight junctions. And that's one of the things that we can do to take advantage of inserting man-made drugs such as liposomes on a nanoscale, because they lack that intimate contact of cell adhesion through protein tight junctions. So this shows us other materials, and again, for prosthetic devices, generally what we would have an implanted material. such as a knee, hip, or tooth here, what we would often want to do is apply a hydrophilic coating. And what that would do is have the ability to adhere an aqueous solution. And once we have an aqueous solution, we can tether proteins on that system, on this particular material. And that protein adhesion is going to give us recognition from the cell. So again, this is our artificial or man-made material. And what we're trying to do is have it perform these tasks, like cell surface identity markers. So we could have this protein on our man-made material so that we could fool the body in thinking that that material belongs there. We could also have tether things like cell adhesion, and this would be necessary, again, for material such as a bone implant. So if we would have like an artificial knee inserted into your femur, what we would want to have at some point is we would want to have bone cells form onto that surface as per se like a glue or a cement. So your body would actually grow bone cells over top of the bone implant that you have in your body, and this would give us a great adhesion and structural rigidity to that implant. So again, these functions of these membranes and proteins are especially important in man-made materials that we would insert in the body. So these last two slides that we just identified have great impact on materials such as prosthetic devices that are used every day for things like knees, hips, dental implants, bone screws, etc. So this shows, again, the wetting, protein, and cell growth regime, to where we would want a material to probably wet. And when that material wets, we can do proteins, and then we would potentially have the ability to have intimate cell contact at that point. So this is where we would want the body to recognize this as something that's naturally occurring within the body. So these, again, show an example of a dental implant. So what we would do is take this tooth or a root of a tooth, be able to put it into the jaw. When it's into the jaw, we're going to be able to form bone onto there so that we would keep that implant rigid. And then what we might do is put an artificial tooth on the end. And again, we looked at that when we looked at nanoparticles, like sintered nanoparticles for zirconium type of dental implants, which would be high strength materials that are active materials and have great biocompatibility within the jaw. And this, again, is an example of artificial knees. Again, we would want to control the wetting and growth of the area on there. Some materials we would like to wet, certainly in protein and cellular growth. These would be identified as areas down here in the bottom. This is where we would want to have bone growth and cellular growth. But in areas like right here, this would act, we don't want to have cellular growth on here. So this could be, for example, high molecular weight cross-linked polyethylene. And in that case, what we're going to do is not have wetting on that surface. Therefore, we wouldn't have cellular growth, and that would keep this relatively smooth, and the water would ride across the surface and act as a lubricant. So that's how we would identify having a man-made cartilage, and this would give us a smooth area where we wouldn't have cellular attack, and we would have high wetting angle, and that wetting angle with the water in between there would act as a lubricant for us. So this is for artificial knees. So again, at this point, that was our look at the water interface for wettability, and this example of water interaction within the body for hydrophobicity, cellular adhesion, and protein interaction. So that ends that part of the presentation. And what we're going to look at next is actually measurement of these techniques. So for example, we might want to take a look at water on the surface, like in our slide right here, water on our surface for our last example, like the artificial knee. So we can tailor that wettability on the surface to tailor cellular reaction of that material. So again, I'd like to thank you for your attention in our presentation, and I'd like to close this out now. Thank you. Take care.